What would it look like if I glowed in the dark? Could we make certain foods entirely allergy-free? Is it possible to bring back extinct species from the dead? Though these ideas seem to just live in the world of science fiction, they are soon becoming our reality. How? Well, with CRISPR. CRISPR isn't a mechanical machine, but rather a modification to the naturally occurring CRISPR-Cas system already used in bacteria against viruses. Before we dive into how the system can be modified, we should first understand how the original process works. Take a bacterium. Now imagine a virus lands at its surface and in hopes of reproducing injects its DNA inside. If this virus was a first-time intruder, the bacterium takes a snapshot of its DNA by adding part of it to its own DNA. Specifically, it inserts it into its CRISPR locus region, a series of spacers which are previously acquired bits of viral DNA from other viruses, and a series of clustered, regularly interspaced, short, palindromic repeats, or CRISPRs. Okay, so why is this snapshot important for anything? Well, pretend that the same virus decides to return. Now, the bacterium quickly identifies the intruding viral DNA and enlists the help of its own DNA to go to battle. First, it creates a protein called Cas9. Next, it translates a section of its DNA that includes a snapshot of the virus's DNA into usable RNA. The RNA and Cas9 then join together and form a complex that quickly finds the virus's DNA ready to clash. In brief, the RNA on the outside matches up with the complementary strand of viral DNA that was previously snapshotted, and the Cas9 acts like a pair of scissors to snip it in half and finally render it useless. Cool, so the system helps bacteria fight against threatening viruses, but how can this make me glow in the dark? Well, imagine if the RNA that's used to guide the Cas9 to the virus's DNA can guide that same Cas9 to your own DNA. Just last year, Dr. Jennifer Doudna and Emmanuelle Charpentier were awarded the 2020 Nobel Prize in Chemistry for discovering this revolutionary new way of modifying the already occurring CRISPR-Cas system. In a nutshell, their alteration to the system essentially replaces the RNA that's originally made in bacteria to fight against viral DNA with a new RNA that's tailored to match with someone's own DNA. Just like before, the RNA guides the Cas9 to a section of DNA to cut it, but now scientists can then insert a completely new gene of their choosing. Yes, like a glow-in-the-dark gene. Although this new CRISPR system can theoretically be used for fun things, its true significance lies in its ability to edit out a range of genetic diseases and even cure cancer. It can even help eliminate certain viruses that can turn pigs into organ donors and insert the genes of extinct species into currently living animals. Although experiments utilizing it so far have only been done on a small scale and it arises many ethical concerns, it certainly has the potential to be a great source for change in the very near future.